morning. I'm Karen.
And I'm Samuel.
And today we are talking about modulation of transferrin receptor by HAV2.
So first we have transferrin. Transferrin is basically a type 2 membrane receptor, which is typically known as a homeostatic. This works in iron uptake, and also it helps with certain things in metabolism. For example, we can see here how iron bonds with transferrin and it goes to an organ, for example, the liver. And it has a weight of 97 kDa and is highly expressed on dividing cells, for example, the erythrocyte.
So now we need to know about lineage. Well, basically we have two types. The difference are the origin that this one it came from chimpanzees and this one it came from Maya Bay. And this one is endemic from west of Africa. They both use the same receptor that are CD4 and well, basically the two it's slower than one.
Then we have our general goal, which is modulation of TFRC expression following transduction with HAV1 and HAV2 cell molecules.
So now we have methods. First, it's Western blot that is used to identify a specific protein. In this case, it was TFRC during levels of the infection. And then we have the cell culture that is also used to make cell growth in a controlled living conditions. In this case, it was used to three different types of cells: HECAT2, NIGE3T, JURCAT, and THP1.
Then we have transfection. Transfection is basically to analyze like a target gene and to produce a protein. This was used in the study, well, first with egg cells. Egg cells were treated using basically an antibiotic, and then we have a plasmid. The plasmid that was transfected with those cells was Y44A. It basically introduced an inactive mutation, which it was HIV that gene. And this resulting cell pellet was then used for Western blot and of course to know TFRC expression, as we can see also here in the image. Right? And then we have the qPCR. qPCR is basically a system to know RNA or DNA expression. In the study, it was used to know RNA expression assessed with TFRC expression. And also, well, it's basically to count molecules. So we can see also here, it was centrifugated at 1000 RPM in 4 minutes.
So now we have results. First, we compare in this graphic the TFR levels in this type of cells, and basically we found that HEV2 it's faster and it grows more than in this graphic that it's about urcal cells, that in the 2 there is slower and less.
Also we can see well the downregulation in THP1 cells because well we can see here that there was a downregulation of HAV2, of course also TFRC expression, basically at a proteomic level at 36 hours and 48 hours post-reduction. This was shown in both of them, but also an HAV upregulation. Then we have basically a TFRC modulation, which was with the plasmid, basically Y44A, which is HIV that gene, and it shows that there is a downregulation of TFRC, which in comparison to the other ones, well, is less than others.
So now we have discussion. Basically we chose 3 different authors, that is Sabaronet, Net, and Reeves, and Marchant. And they basically the first one talk about how are the levels in the intracellular iron in HAV infection, and this is agreed with the study. Then we have Net that talk about that the superinfection resistance are observed in most of retrovirus, and this is incorrect with the study. And then we have this one that it talks about that TFRC upregulation have different differences in the HEK293 T cells and JURCAT cells, that we could see it in the graphic. There is this one and this one, and so that it's correct with the study.
Then we have the conclusion. The first conclusion is with the TFRC expression. This shows how certain methods, for example, qPCR or Western blots, are used in medicine and molecular biology to know also the expression of certain genes, as we saw here with the RNA expression. Then we have the HIV influence on iron uptake, and we can see how certain pathogens influence also in, well, certain things, for example, with the iron homeostasis and certain metabolism process, for example, with the liver and things like that, also using the qPCR and Western blot. And finally, we had the HIV2 impact. This works in microbiology or also in molecular biology to know like the difference between, well, HIV1 and HIV2 in this case, and also to identify how this dynamics changes with this and the other one, and how the transferrin receptor changes too.
So this one for today. Thank you for watching and see you in the next video. Bye bye.